One of the best bits about being the chair of the BSDB, the best bit of my job, is handing out these awards. And we have 4 now, actually. There are only 3 up here, but we've got a brand new 4th one that I'm going to hand out. And of the 4 medals, I get to hand out 3 at the meeting. And the first one I'm going to hand out is the Tickle Medal. This is Cheryll. My pointer's not working, but that is Cheryll. She's one of my heroes. She's the reason I went to UCL. She's a superstar, and she would have come actually today, but she has a 99-year-old mother that she's nursing in Exeter, and her husband's not well. But she has recorded me a little video. So what the Tickle Medal is for: mid-career, kick-ass female developmental biologist, essentially. And it's a pretty nice medal, which I haven't lost. It's here. And unfortunately, I met the person who's going to receive this medal last week in Cambridge, and she was coming. And then on Friday, she sent us an email and said her daughter's sick with COVID, and so she daren't come and be the super spreader. But I'll give you some clues who she is. So if you go to Cambridge, and I was in Cambridge last week, and you want a beautiful picture of a human lung, you go 2 places. Either the Wren Library, and I go there a lot because they have beautiful antiquarian books. I collect antiquarian fish prints. And this is not Vesalius, because Vesalius was nicked, but the next best, you can see a beautiful picture, antiquarian, of a lung, human lung. Or you can go to Emma Rawlins' lab. So these are, I can't point, my pointer's not working, but these are human lung sections. And I use them in my, oh, thanks. I use them in my medic undergraduate lung development lectures. They're outstanding. She works on development of the lung in mouse, in human. She uses organoids as well. Her work is lovely. And it's moving from developmental biology into regenerative biology stroke medicine. So she really, really deserves this medal. But I'm going to let Cheryll do the talking because it's her medal. And here she is. They promised me this would play. I'm going to go back. Can you make it so it does play? You need, we practiced and this did play. There's a guy at the back that did something in rehearsal. I'll play it one more time. Now you really need to hear her words. She and I got up at 6 in the morning to record this so that we could, before her nursing chores.
Hello, Emma. Congratulations. I first became aware of your work on mouse lung development during your postdoc with Bridget Hogan. And since then, you've certainly built on that wonderful start. Your work has illuminated how the mouse lung is built and how it is maintained in the adult. And this has made really quite a big impact on thinking about ways in which one could possibly repair a lung. Your recent work on human lung development is particularly relevant and exciting. And now you're poised to move from a description of the human lung to an experimental analysis. And I look forward very much to seeing the latest chapter in your research.
Okay, so here is the medal. And if Emma's daughter didn't have COVID, I would physically hand it to her. But Emma, if you're listening, I'm coming to Cambridge in the next month and I'll bring it to your lab. It's a beauty, you'll like it. Now, Emma is going to zoom in, so I'm hoping that you guys are cool for this.
Hi, Paul.
Yes.
Is that working?
Yes, it is. I'm going to walk off the stage.
Super. I'm trying not to cry after hearing Cheryll and failing to share my screen. Here we go. Okay, I hope this is working. I am beyond honoured to receive this medal. Thank you very, very much. And I'm beyond devastated not to be here in person to come to the bar afterwards. But one of those things. I didn't want to receive a medal and repay with COVID. I thought that might be wrong. But it was a pleasure to put this lecture together. One thing that I was asked to do was to give a little bit of a career perspective. And this was actually, turned out, I was a bit scared, but it turned out to be really pleasant. We're always thinking ahead to the next big question, the next grant, the next paper. And it was really pleasant to look back and see, oh yeah, and actually continue a trajectory of the questions we've been asking and why we've been asking them. So I started my scientific career, this works, back in 1998, not even in 2000, in 1998 as a PhD student in Andrew Jarman's lab in Edinburgh, working on sulfate specification in the peripheral nervous system of the fly. And in particular, I was tasked with sorting out some enhanced and suppressed alleles from a screen that the lab had done. And this is one of those, a gene called echinoid, which turned out to be a cell surface molecule. And you can see echinoid has a rough eye phenotype, meaning this beautiful normal wild type pattern mormatidia is disrupted. And when we looked at the eye disc to see what was wrong, it was immediately apparent that we had R8 twinning. What does that mean? Well, this is the morphogenetic furrow that spreads across the eye disc, and in its wake, single individual photoreceptor 8 cells are specified. You can see they're specified in a beautiful pattern. And in the echinoid mutant, we had pairs of photoreceptors. So we looked at this and we thought, this is going to be a notch phenotype. That's what will be wrong. We were completely wrong. It turned out to be an EGF receptor phenotype. We had overactive MAP kinase signaling. And the first thing the R8 receptor actually does is signal via EGF receptor to surrounding cells to recruit the next photoreceptors. And this was what was going wrong in our mutants. So we published this back in 2003. And echinoid also had a bristle phenotype that we published the following year. And that turned out to be notch specific. So at the end of my time in Andrew's lab, we almost certainly went for a lab picnic, as that's how we celebrated. I'm sure they were delighted to get rid of me and delighted to move away from the fly eye. But it was a fabulous place to work, but I was anxious to move on. And obviously the end of your PhD is a great time to do that. And I moved to Bridget Hogan's lab to learn mouse genetics and mouse developmental and cell biology. So for those of you who don't know Bridget, she was really is a pioneering mouse geneticist who works on all sorts of different aspects of mouse development, but particularly embryonic carcinoma cells and EG cells, as well as the developmental biology. So when I joined her lab in 2004, she just moved to Duke to be chair of cell biology. And she said to me, Emma, you can work on anything you like in my lab, but it has to be in the lung. We're going to focus now, and we're going to focus on lung. So looking around at the developing lung and talking to Bridget, we came up with a bunch of questions, but just briefly to remind you, because the only time we've thought about your lung recently is to try and think not to get COVID, to put your mask on. Well, of course, your lung has two main regions, the conducting airways that the air moves in and out of, and the gas exchange region, the alveoli. So the question that we focused on after many discussions with Bridget was, what's the stem cell hierarchy in the mouse conducting airways? And in particular, I wanted to know, is it the same in development as it is at homeostasis, as it is following injury? And look at this question now, and then the last week putting this talk together, in some ways this seems a little bit of a trivial question. What do you mean you didn't know this? But we didn't know when, and we didn't know that it wouldn't be the same or it would be different. So this was a really exciting time to be working on this question. And what approach should we take? Well, there are three major cell types in the mouse airways, the basal cells and the trachea, the club cells, which are found throughout, these are secretory cells, and of course the multiciliated cells that we've just been hearing about. So the approach we decided to take was to lineage label the three different types of cell, and I made different pre-ER animals the basal cells, the club cells, and the ciliated cells. So you can see here, I've got 3 little stories to tell, one about each cell type. And I was wondering, what should I talk about for this talk? And of course, I have the privilege of following John. So it had to be that I decided to share you a couple of interesting facts about multiciliated cells in the airways. So multiciliated cells, the question we're asking back in 2005 was, do multiciliated cells divide during airway development, or particularly during repair? And some of you are probably thinking, this is a crazy question. Why on earth did you ask this? So you all know that multiciliated cells endoduplicate their centrioles. They get docked at the apical surface of the cells to form the basal bodies. We get lots of cilia. How can these cells with endoduplicated centrioles possibly divide? That puzzled us as well, but there was lots of reports in the literature that they could. And in particular, a report from lung regeneration, lung repair, that following injury, ciliated cells could divide and could probably act as stem cells. So we really felt we needed to find some clarity to this, and hence the lineage tracing. So colleagues at UNC Chapel Hill had just cloned a FOXJ1, human FOXJ1 promoter, and made a FOXJ1 GFP mouse. You can see this is simple dissecting scope images. It beautifully labels the ciliated cells. And I looked very hard and we could never find them dividing. But what about if you really injure these cells? So we had to do our lineage tracing. So we made our FOXJ1 pre-ER. And looking back on this, I remembered actually, I used to do all of my experiments in duplicate. So once by crossing to the Rosa 26R LAC-Z, that gave you wonderful spatial resolution on the dissecting microscope. You could really see the patterns of labeled cells. Then again, same query, but using a Rosa 26R YFP in order to be able to do decent antibody staining and get on the confocal. So I'm going to show you some LAC-Z images now. You can see without injury, we have these ciliated cells, lineage labeled, sitting within the airways, and these are taller cells are the club cells. You hit your mice with naphthalene, which kills all of these cretory cells. And the first interesting fact I want to tell you about ciliated cells is that they have a really plastic phenotype. And actually, the ciliated cells survive this injury. They squamate, they become really long. And presumably they do this in order to maintain the barrier epithelium, maintain that barrier surface. But if we look 3 weeks later, the ciliated cells that were labeled still persist. They remain ciliated. We could find no evidence that they divided and no evidence that they made any other type of cell type. So they look like they're terminally differentiated, but they survived the injury and contributed to the repair, presumably by the virtue of being there. Interestingly, if you look at 100 days, about 3 months after the injury, then instead of being nicely distributed throughout the airways, our labeled cells are now grouped into patches. So really suggesting that an unlabeled cell type in this region has divided, acted as a stem cell, made a load of new cells, and have pushed our ciliated cells together. So we did this with a second injury, just to be really sure, but we could find never, ever any evidence that most ciliated cells could divide during airway development or repair. But I love this plasticity in their cell shape. So we published this back in 2007, and it really led to another question, that if we have these wonderfully long-lived cells that survive the injury and 100 days later are persisting, how often do they actually turn over? So instead of thinking about protein turnover within a new vesicle at the lifespan of nanoseconds or something, we're now going to think about the lifespan of an airway ciliated cell at homeostasis in the airways. So when I say homeostasis, of course, I mean in a mouse, a sterile mouse, in effect, breathing filtered air. So in order to answer this question, I lineage labeled a bunch of FOXJ1 pre-ER Rosa 26R YFP mice, analysed a few of them to say how many ciliated cells were labelled. Do I only have ciliated cell labelling? Then I put the cages in the corner of the animal room and left them, and sampled over the next 18 months to ask how frequently do I see lineage label ciliated cells. These are tracheal sections, and you can see we have fewer cells labelled at 7 months and only a rare number in the trachea at 18 months. So plotting this data out, what I wanted to do was make an exponential decay curve like you should see or you do see in a textbook, and ask, can we predict the half-life of a multiciliated cell? And this worked beautifully for the trachea. You see our exponential curve fits really well. And we can estimate the half-life was 6 months. So meaning if you lineage label the cells, then 6 months later, about half of those will have turned over, but half will still persist. In the bronchioles, the curve fit less well, but it does fit. And we estimated the half-life at 14 months, so well beyond the lifespan of many normal wild-type mouse certainly living in the wild. So my original plan for this work was to roll it in with the work I was doing on secretory cells, and actually the secretory cells are the stem cell that replaces the ciliated cells, and publish it all together. But it didn't really fit in that paper, so we ended up publishing just in one of the lung journals, one figure, two table paper about ciliated epithelial lifespan in the mouse trachea and lung. So I was happy with this. I had my exponential decay curves, and I thought it was really important baseline data for future experiments. But something Bridget always said was that, Emma, you have to publish everything because you never know who's going to be interested and who your work will be useful for. Well, to our surprise, actually, this paper has almost 300 citations because it's one of the few estimates of ciliated cell, or actually turnover of any airway epithelial cell. And this really matters if you're working on cystic fibrosis, for example, and are interested in gene therapy. I actually get a surprising number of questions about this work, even now, more than 10 years later. So I want to show you a final bit of work from the work we did in Bridget's lab, and that's to ask a slightly different question. So in normal development, when we haven't built the airways yet, we don't have these different cell types. Is there a multipotent epithelial progenitor population that gives rise to the entire epithelium of the lung, so both the conducting airway cells and the alveolar cells? So we hypothesized that this would be located at the distal branching tips based on previous work that Bridget and others had done. And it won't surprise you to hear that I've generated an ID2 Cree yarn mice to lineage label these cells, and indeed showed that they both self-renew during normal development and make the airway cells and the alveolar cells. So we submitted this paper to development for review in 2009. And a few weeks later, Daniel was born. So this is Daniel. Daniel is now almost 13 and has COVID, the reason I can't be with you today. And here he is with my good friend, Janwen Q. So I was very lucky. The reviewers asked for very few new experiments during the revision process. And I was able to bring Daniel into the lab, and it was handed round, cuddled by some very broody postdocs in order to let me finish this work and wrap up my time in Bridget's lab. So I'm really grateful to Bridget and the entire lab for the fabulous time I had there. And it was a privilege to be able to organise essentially a retirement symposium for her in 2018, and many of us were able to come back and celebrate. Though I should say she may have closed her lab, but she's certainly not retired. She remains extremely busy. But back in 2009 then, I moved with my family to the Gurdon Institute in order to start my own lab. And I was very fortunate to have an MRC Career Development Award. I remain incredibly grateful to the MRC. So 2009, if you can put your mind back that far, we occasionally used Skype. We certainly didn't have Zoom. I had a 4-week-old baby, and I wasn't doing very well at coping with him. And I certainly wasn't in a fit state to fly across the Atlantic for fellowship interviews. The MRC arranged everything online, gave me the grant, and let me start. And I remain incredibly grateful for that. So the question we wanted to ask was, how is airway homeostasis maintained? Digging in more detail into the lineage, but also to begin to ask about the signalling and the maintenance of this structure. And the work I haven't showed you, that we had a model pre-existing, which said actually it's the basal cell, which is a stem cell. The basal cell makes club cells. The club cells can also divide and self-renew, and they can generate ciliated cells. We didn't know if this could ever happen. So we wanted to dig more into the lineage of this, and the specific questions I wanted to ask was, are the stem cells heterogeneous, and what are the stem cell dynamics? And Julie Watson, my first postdoc, took this on. So we collaborated with Ben Simons, in particular Stefan Rawlins, and Ben had just had a paper a couple of years before, many of you will remember it, with Phil Jones, really pioneering work showing that by looking at clonal lineage labeling the skin, they could begin to do some modeling and pull out the stem cell dynamics. And I wanted to do this for the trachea. So we started Julie started lineage labeling cells in the trachea, and the trachea is really nice. You can cut it in half, open it up like a book, and image the clones on the internal surface. So unfortunately, my animal license, the Home Office, wouldn't let me do injury. So we had to do homeostasis, which meant these were very long experiments. But we got to work, and Stefan and Julie came to the conclusion there was more than one type of basal stem cell. So how did we investigate that? Well, actually, at that point, then I went to Bertie Gottgen's lab and did some experiments that we'd never dream of doing now. But Bertie Gottgens and his PhD student, Adam Wilkinson, had set up single-cell QRT-PCR. So of course, now you just sequence the whole damn thing. But we had to pick our favourite genes, and we had examples of a few of them that did seem to be heterogeneously expressed within the basal cells. So another collaborator, Cedric Blampane, gave me some mice so we could do more lineage labelling and test our hypotheses. So we managed to publish this in 2015, which to note the gap between starting the lab in 2009 and getting the first paper out, it was quite considerable. But we got this to work. And the second question we were asking at the same time was, what are the signals that regulate steady cell states turnover? What are the signals that regulate the turnover of this structure? And we decided to focus on FGF receptor signaling. So my first PhD student, Guy Balasouria, worked on both FGF receptor 1 and FGF receptor 2. And I'll just summarize a little bit of that now. So Julie and Stefan had worked out our hierarchy, showing that indeed we had two types of basal cells, and that one of those essentially fated to differentiate and to become a secretory cell. And you have these predominantly asymmetric divisions. So Guyan was able to show that actually the rate of cell division of this structure was beautifully under control, and it was actually actively repressed at steady state by active FGF receptor 1 signaling. So in the FGF receptor 1 mutants, the rate of turnover increased and cell flux through the system was faster. We had a more conventional phenotype with FGF receptor 2. Here, the FGF receptor is actually required to maintain the stem cells, and in the absence of FGF receptor 2, you pushed into the differentiation pathway, and everything differentiates to the secretory cell phenotype. So we'd answered a lot of our questions, well, many, many more questions always. We'd answered a lot of our questions we started with about the stem cell hierarchy in the conducting airways. At some point in this process, Lizzie was born. Well, in 2012. You can see, unlike Daniel, who was handed around the lab and generally cosetted and petted, Lizzie was dumped on the floor as a second child while we got on with things. And I just want to highlight another paper that's very minor, but we're extremely proud of. This was our 5-baby paper. So between this work, actually, I should say 6 babies, between this work starting and actually being published, I had 2 children, Marco had 1, and the first author had 3 during her PhD. But we got it out there, and I think this contributed to Jo getting a senior lectureship when she left and moving on to teach at Anglia Ruskin University. But the other question we maintained our interest in was lung development and the potency of our multipotent progenitors. So I simplified earlier, and I told you that tip cells were multipotent, which they are, but they're actually sequentially potent. So initially, they make the bronchiolar cells, and at later stage of development, they give rise to alveolar cells. And the question was, how do you switch from one to the other? So this was addressed by Asura and Marco, and essentially the cells read a local level of glucocorticoid signaling, we think, and this somehow, we still don't know how, interacts with STAT3 in order to control their internal fate and ultimately the fate of the progeny. But this really started us thinking about lung development over time. And in particular, we realised at this point, we could get hold of human embryonic lung samples from the HDBR. So if some of you are not familiar with the HDBR, this is a fabulous tissue bank funded jointly by the MRC and the Wellcome Trust, started by Andy Kopp, and they are really the best tissue bank ever. And via them, we can access human embryonic samples from 5 weeks to about 22 weeks gestation. So this is a 7-week-old human embryonic lung. And this is a section of that lung. We can see our favourite tip epithelial cells, that are the progenitors. These are the ones that are growing out and undergoing branching that give rise to differentiating cells that are left behind. So Marco Nikolic was a PhD student to ask the question, how similar are these human structures and human gene expression patterns to what we're so familiar with in the mouse? And at the time, we did some very simple things. We dissected multiple populations of bulk RNA-seq. We compared that with our mouse work. And we found enough differences that we decided we wanted to make a human-specific model that we could do functional biology in. So Marco started trying to grow organoids. So he was dissecting off our tip cells that we knew were multipotent, plating them in matrigel, and our favourite growth factor containing medium has got lots of FGF and Wnt, and it inhibits BMP and TGF-beta. And in these conditions, we can expand the cells in a self-renewing state. Actually, in this first passage, if you dissect off one of these, it looks remarkably like starting material. So they expand beautifully, they're maintained undifferentiated, we can freeze thaw them, and in the right conditions, we can switch to airway or alveolar fate. So this is very much like, in my mind, an embryonic stem cell-like line, but now specific for the human lung. So Marco published this in 2017, and he now has his own lab at UCL. He has a clinical fellowship and is continuing to work on human lung development. But as we were bringing this to a close, this little story, I was starting to write my senior fellowship applications and decided that I would take what felt at the time quite a scary and bold decision, that we'd have everyone in the lab doing the human developmental biology. It seemed that this was such an exciting time to be doing it, and we had a system that we wanted to exploit. Let's put everyone onto it and see if we can make some sense. And of course, the advantage of this was if it didn't work, we'll be able to backtrack pretty quickly and switch back to the mice that we know and love. But the big concern I had at the time was this is our first real foray into in vitro biology. And how did I know that what we'd see in the dish, in our organoids, the functional genetics we wanted to do, would really reflect what was happening in vivo and be interesting or important to anyone? So we decided we have to spend a lot of time flipping backwards and forwards between describing the human structures and doing our functional work in the organoids in vitro. So that was the approach we took, and I'm very grateful to MRC for funding it. And I'll just tell you a few things that we've been doing in the last few years that we're now trying to get published before I give you a little bit of perspective of where we want to go. So Darway's son was a PhD student in the lab. In a couple of weeks, he'll be moving to Boston to start a postdoc abroad. And he wanted to ask the question, what's the genetic network that controls the self-renewal of these tip cells? So we really wanted the ability to be able to do intrinsic, cell-intrinsic genetics to manipulate gene expression of these cells, rather than just changing factors in the medium that we thought was quite boring. So Darway set up multiple genetic tools. He developed knock-ins, his system we called organoid EasyTag, inducible knockdowns, somewhat tweaking the CRISPR-I vectors to get them to work well in human organoids, and inducible overexpression. We put these into AdGene at the time that we pre-printed the paper. And I made my year last year, really, when AdGene said that my lab had hot plasmids. It's not something I ever thought I would see. And we published the technical part of Darway's work at the middle of last year sometime. But the key question Darwin really wanted to ask was a functional question, which transcription factors are required for the tip progenitor self-renewal? And having the CRISPR-I in hand, he was actually able to do a screen for self-renewal phenotypes using the organoid. So a multiplex screen, a pooled screen, only 49 transcription factors in this case, but now this has worked so well, there's a potential to expand this, and I think a lot of possibilities have come out of his work. But the highlight of what he saw was multiple genes affecting self-renewal of our cells, and we decided to focus on SOX9 to really pin down its mechanistic effects in the human progenitor cell types. So simultaneous to all of this, Kung-Tae Lim in the lab has been asking the question, how do human lungs coordinate alveolar development? So just like the mouse, there are multiple phases of lung development. So initially the lung branches away to make airways, and all our published organoids were from this airway differentiation stage. And at later stages, it switches. We still have tip cells, but now they're generating alveolar cells. So of course, we want to know about the difference between these stages. How do you switch from airway to alveolar development in the human? So as part of our single cell work, we noticed that our tip cells were moving quite a lot within our UMAP, and our later stage tip cells are very close to the alveolar type 2 and the type 1 epithelial cells. So Kung-Tae started growing these and really showed how you acquire both alveolar fate and differentiating competence in the human lung. And as a nice side effect, we got self-organizing organoids for the first time, which again, you can find on bioRxiv. So I'm going to end here. I'm sure I must be out of time, and just very briefly share what's ongoing and what's next. So we're continuing to work on things like signaling mechanisms, controlling human cell fate and differentiation. We've actually done a small arrayed screen and have that arrayed phenotypic screen that we have working quite well for type 2 cell differentiation. We're starting to do lineage tracing in human lungs. We've been working on this about a year, and I think really the last few weeks I've just got my head around just how complex a problem this is, but a very exciting one. We've moved our CRISPR work on to now using base editing for modeling of specific diseases, and have evidence that this is going to work. We're starting to look at some more physical factors in collaboration with various different collaborators, their effects on lung development. And finally, we have some interesting cells in the human embryonic lung that we found with our collaboration with the Type 1 lab and the single cell axis work we've done that just don't exist in the mice. So how are we going to work out what they do in any sort of physiological and meaningful way? So we're going to have to work on what will be new model organisms for us, but of course, historic model organisms from the field. And we're starting to think about whether we should use rats or sheep, or the best way of exploring some different types of lung cell biology in these animals. So I just need to end with a thank you. I'm incredibly grateful for everyone who's come and worked in my lab over the last few years and made it a very fun place to spend time and be. They're all listed here, as well as our many, many collaborators who've helped us with lots of different aspects of these projects, and all of our funders, primarily the MRC, who have been absolutely fabulous, but also the Wellcome Trust and various others. So I'm going to stop here. I hope you can all hear this. The usual Zoom thing of you never quite know, but that's me done. Thank you for listening.
We could hear you, Emma, and it's a fabulous talk. I was going to look to Sally to check out whether on these occasions we do have questions. You think we do? Have we